Good afternoon, everybody, and it's a great pleasure for me to welcome you again to this second in the series of our celebration lectures. And as most of you know, today it's Professor Melita Gordon who is going to give us her a lecture. But before Melita takes the floor, I just would like to extend a special welcome to the principal of the college and the Wellcome Trust Governor and Dr. Chu, who are visiting MOW at the moment, and that really just enhances this occasion and brings it to a different level. So you're all welcome, and I hope you enjoy this afternoon's lecture. So a few words about Melita. Melita is no stranger to most of you here, but you know her as the Typhoid lady, probably. But actually, Melita is a clinician scientist. She's been associated with the MOW program for nearly 22 years now. She started her research career pretty much here back in 1997. But since that time, her work has really focused on trying to understand the salmonella infections. So typhoid salmonella in HIV-infected individuals, but also more recently, typhoid fever. And most of you will be aware of her landmark studies here, especially the Typhoid vaccination campaign that went around a few months ago, but also Strata and other studies. But she's the most senior academic from the University of Liverpool, and has worked here as a Wellcome Trust Training Fellow. That's how her career started. But her career has been stellar in that she's made a lot of achievements during this time. And this was recognized in 2015 when she was promoted to professor at the University of Liverpool. And I think for me it's a great pleasure to invite her to speak to us, because this is really in recognition of her recent Great Award. She was recently successful in obtaining the National Institute for Health Research professorship, which is worth £4 million. And that's no mean feat. It's only really special people who get that award in the UK. So Melita, this is a fitting tribute to your achievements, and I'd like to invite you to give your lecture. Thank you.
Thank you very much, Henry. Thank you for piling on the pressure. Thank you for all the years of sharing offices at MLW. I think everyone can hear me, can they? So thank you for coming, everyone. It's wonderful to be up here. So I believe at these lectures it's traditional to give a slightly more personal view, tell a little bit about your own story. I thought I would talk about decisions and people and what's unknown, as well as your science. So I will try and do that. And as I was thinking about giving this talk, it occurred to me that I'm 53-year-old, I'll save you all from doing the maths. But as I thought back, I realised you really only have to make about one major decision a decade. Well, that has certainly been the case for me, and I thought that might be a nice way to hang this together and provide a little bit of entertainment for you as well. So, this is me being born. Of course, that's not a decision I took. You don't get any choice in that, do you? So I was born in London. This is my mum. She was a junior doctor at the time. The reason she's so glammed up is that she's posing for a photo for my dad, because my dad had to leave the UK to go to America and find a job, and she was going to follow with me after I was born. So here's my very glamorous mum with me newborn, and here's my dad, who I first met when I flew to America and I was about 6 weeks old. And you'll see that he was waiting for me with a little panda. So this is the panda. That was my first house move. That was my first house move. I estimate I've moved house about 20 times, and Panda has gone most of the time, so I thought I would bring him as a visual aid. You can hand him around, but I do want him back at the end. Okay. So I was given the name Melita. I was named for my mother's mother. This is my grandma. She was actually my favourite grandma. She was a history graduate from the University of London. She was one of 5 children. She went on to have 5 children herself. And she had the name Melita. She in turn got that from her great-uncle, who was working on the island of Malta. Now, the Latin name for Malta is Melita, and he was working for the Royal Navy, building a ship called HMS Melita. So I'm named for an island, I'm named for a ship, I have a little bit of heraldry and a Maltese cross to my name, which is nice. But not only that, Paul the Apostle was shipwrecked on the island of Malta in a very vividly described shipwreck, which I'm pleased to say everybody survived. That's good for my psyche, you can imagine. And subsequently this hymn was written, which is For Those in Peril on the Sea. It's a very famous hymn tune, and the name of the hymn tune is Melita. So I'm not only named for a ship, but also for a shipwreck, and I have my very own hymn tune. So I've got a great name. And I grew up as a little girl in California. It was very sunny, it was a very happy childhood. When you're a little kid like that, you don't get the choice to make many decisions, do you? But you do get to name a few things. So I had a panda, which I named Panda. And I had a baby bunny, which I named Bunny. And I had a tiger tabby cat, which I named Tigger. So I think we are drawn to the inexorable conclusion from the data that I don't have a natural talent for deciding on interesting names. We had moved to America for my dad's career, but we moved back to the UK in 1973 for my mum's career. She wanted to move back into medicine. So this is me, my two sisters. They both have a great deal more imagination than me. And my mum re-entered a career in medicine, and I think there's no doubt at all that my two sisters and myself were her greatest career supporters, and it was that experience of seeing her enter into her medical career and embark on training that made me decide to be a doctor. And I think I probably made that decision very quickly in 1973. So this is me quite a bit later. Definitely decided to be a doctor, but not only a doctor, I decided very quickly I wanted to be a doctor in Africa. Now, I only knew one person in my whole life who had been in Africa, and it was this man. He was our priest, Geoffrey Harris, a wonderful gentleman who had a very great impact on me. And he has since died. This is the stained glass window in our local church to him, a missionary, teacher, priest, physician, and lepidopterist, which means someone with an interest in butterflies. So this decision has always had a big part around my faith and a sense of vocation around both medicine and working in Africa. And of course, anyone in medicine knows that once you've decided to be a doctor, you basically don't have to make another decision for 20 years. You apply to medical school, you get in, you do your training, you do your junior doctor jobs. That took me about 20 years. So I didn't really have to make any more decisions after that early decision in 1973. But I did have one other decision to make. So I went to Cambridge to study medicine. In 1984, this dashing young man with a very interesting taste in head adornment, shall we say, asked if I would like to start going out with him. And I said yes. Big decision. I didn't know how big a decision this was at the time. I didn't know that I had met the person who was going to be the greatest supporter of my career and have more confidence in me and my ability than I did myself. So we graduated, we got married, and here on the right is a picture of what happens if you spend too long with your heads together for about 30 years. A tree grows out of it. I think there has to be a metaphor in there, but I'm not quite sure what it is. So I started training in medicine. The next 10 years passed in a kind of blur of exams and on-call, both at medical school and in junior doctor jobs, through 6 different cities, and I also managed to accrue a bit of international experience in Kenya, Chile, Zambia, and we worked in a reconciliation community on the Peace Line in West Belfast. And at the end, you come out with this sort of alphabety spaghetti list of qualifications after your name, and you're all equipped as a kind of basic jobbing doctor to go and be a physician in Africa. My family has a bit of a thing with buses and bus stops. My dad proposed to my mum while she was standing at a bus stop waiting for a bus. He was so terrified he waited till the bus was nearly there and she didn't have time to make up her mind. So I had a similar kind of experience in 1993. Suddenly, a whole bunch of decisions came upon me, having been trugging happily for 20 years without having to think about anything very much. In the UK, we kind of say buses are like this. You can wait for hours and hours and hours, and then suddenly 3 buses come at once. And that's what happened to me. I had 3 big decisions that I made in 1993. The first was that we were going to start a family. So these are our 3 kids. They came quite closely after each other, Tim, Anna and Miriam. They spent a lot of their childhood in a rather grubby state, being dragged on overland trips through Africa. They had a wonderful and very formational time in Africa, and they scrub up quite well as well. And again, at the time that they were born, I did not know that I was meeting my next 3 greatest supporters of my career. Second decision was the decision that I definitely wanted to go into research in my career. And this was largely because of an experience that I had working in Zambia. We were in a bush hospital and had the experience of cases coming through the front door. We were in the early days of HIV in Africa. And I think Stephen and myself both had a strong sense that if we were going to work in Africa, it would not just be clinical practice. We needed to be very involved in either teaching or research or both in order for things to change in the future. That was a very important experience we had there. And ultimately, it meant that we ended up in Liverpool and we ended up in the clutches of this charming trio of gentlemen, Peter Winstanley, Malcolm Molyneux and Michael Chu. This was in the days when MLW was very, very first being set up with the Wellcome Trust. And it was ultimately the path through which I got my Wellcome Trust training fellowship, which was really the start of my research career. And the third big choice I had to make, having trained in general medicine as a physician, was my specialty. And I picked gastroenterology for a combination of reasons. One was that I quite liked the idea that, unlike infectious diseases, it would give me a practical skill, endoscopy. One was that I met a number of wonderful gastroenterologists who I really admired in my training. And the final reason, I have to be honest, was that these wonderful gastroenterologists flattered me and told me I'd be good at it. So I thought maybe I should do that because no one had told me I would be good at anything else in particular. So I ended up training in gastroenterology. That was a 14-year training in clinical medicine. And much as before, having made those decisions, the next decade unrolled under its own steam. We had 10 years of raising young children, more on call, both in the UK and Malawi, and work towards a doctoral thesis. And the majority of this time was spent in Malawi. I was trained by some wonderful people, Dermot Gleeson and Martin Lombard, both Irishmen. I seem to have a thing for Irishmen. Trained me as a gastroenterologist. And Rob Reed and Tony Hart were my academic supervisors, my PhD, and they trained me in research. Tony Hart, who's Professor of Microbiology in Liverpool, a few of you may know, he died right towards the end of my PhD. He never actually saw my thesis written up, but he was a wonderful and very kind and generous supervisor. And I'm very thankful to all of them. 10 years in Malawi, and this was the time when I really first posed my very own strongly formed research question, I would say. We set blood cultures going at MLW in 1997, and I was home one lunchtime, and having picked up the kids from school, and I got a phone call from the lab. They were really excited because there was a positive blood culture. Can you imagine? There's only one from the lab here. Can you imagine that now if you phoned every time a blood culture came positive? I got a phone call, they said, we found something. Salmonella typhimurium, and I distinctly remember having to think, what? And I kind of rummaged through my head and thought, now typhoid, I know, that's a proper tropical disease. Healthy people get it. It's caused by typhi and paratyphi. And I thought, typhimurium, shouldn't that give you diarrhea? And I went away and looked it up a bit, and vaguely was able to get the impression that it sometimes was in the bloodstream with people with HIV. It might be recurrent. That had been described. But it really got me thinking. This phone call one lunchtime. What was this bug doing there? Why was it in this particular patient? How had it got there? If it was going to be recurrent, where was it hiding away? What was the pathogenesis? So I made some really expensive international phone calls to some international experts in the UK and America. Stephen still worries about the cost of those. I'm sorry about the phone bill for that. But the answer from all of them was they'd never heard of it, they didn't have the faintest idea, and they really couldn't answer my question. Which I'm just going to take a little moment to say something about. I think it's no secret that the Gordons quite like an adventure. Right from my childhood, we've done overland trips. I did 2 big overland trips in the States. We've done big overland trips with our kids across southern Africa. When I started my fellowship, we drove from the UK right through the Middle East and down Africa to Malawi. We went on family holiday in Afghanistan in 2007. We quite like an adventure, and we quite like that sense of journeying and travelling to somewhere new and somewhere unknown. And I think there is an important parallel here in science. So those of you who are my students, and I'm sure those of you who are other people's students here in MLW, are always told you start a study with a hypothesis. And you have aims that you wish to frame in order to prove your hypothesis, and you design objectives which will allow you measurable ways of proving this. And sometimes that all works fine and dandy, and your experiments show exactly what you expect. But sometimes it doesn't work out that cleanly. The experts don't know, your hypothesis seems to be wrong. If you're me, your hypothesis has been completely upside down sometimes. The data don't quite fit. Your experiment doesn't seem to work. You can't find any published literature about this. Your supervisor shrugs and kind of goes, oh, no. And that may be because you've described your experiment, you've designed your experiment badly. It may be because you've messed your data up. It may be because you've made a mistake in the analysis. It may be because you've made a mistake in your protocol, but it might just be that you've stumbled on the most interesting finding you will ever make. So please, never try and gloss over irregularities in your data or anything that doesn't seem to fit. It might be the most exciting moment in your career, so don't be afraid to ask questions if other people don't seem to know the answer. At the end of my time working in Malawi, I had described a bimodal pattern of age incidence from the blood cultures that we were doing, and we had described that this NTS bloodstream infection happened in children, and those were children with malaria or HIV and malnutrition by and large, and then there was a second age peak in adults with HIV. And I persuaded someone called Charlie Gilks, who I think now works for WHO, to give me a very small amount of money. He had a little bit of money left over in a grant, and he gave me £6,000 to recruit a cohort of these patients, adults, and follow them up to see what happened to them, because I wanted to know what their outcome was, and particularly did the bug hide away and recur over and over again. That seemed to me very important in the pathogenesis. So I recruited 100 patients, and what I found that half died as an inpatient, 3/4 were dead in 1 year, and then on the bottom graph, of those who survived, nearly 50% went on to have a bloodstream recurrence of NTS, and we were able to show that it was the same serovar and the same bug coming back over and over and over again. So it seemed that my thought that the bug was hiding away and recrudescing was most likely correct. That set of observations was enough for me to go to the Wellcome Trust, very much supported by Michael, thank you, and get my first Wellcome Trust clinical training fellowship to do some further investigations. And this is my first research team. Ansted Gershom, Peter, Dan and Medrick, at least 2 of them are still working for MLW. I don't know if they're here in the audience, but they were a terrific team. And together we were able to prove in a more definitive way by taking blood and bone marrow samples and doing quantitative culture of them with pour plates, both when the sample was unlysed and when it was lysed. We could show that we increased the yield by bursting the cells, both in blood and particularly in bone marrow, which showed that it's hiding away intracellularly, replicating in the bone marrow, very persistent in adults with HIV. And that led to a large number of studies really over the next 20 years, describing in a lot of detail the immune defects in these different susceptibility groups in adults and children. So that included work at the gut mucosa, showing that there is a deficiency of IL-17 producing CD4 cells early in HIV, and that live Salmonella challenge produces cellular immunity in the gut. Because of this capacity of the bug to hide away inside cells, we looked at cellular immunity a lot, both at the inflammatory environment that allows the bug to persist inside macrophages, and also that the cellular environment around CD4 and CD8 cells that supports both the killing of bugs intracellularly and the subsequent development of antibody defences. And finally, we looked at antibody defences and found that they were deficient in children, but counterintuitively, there was an excess amount of antibodies produced by adults with HIV, which is a very surprising finding to us and has a very important implication that trying to develop a vaccine based on antibodies that you want to give to adults with HIV could be problematic, because if anything, the problem is they have too many antibodies rather than not enough. So these are very important pieces of work. I'd like to draw the attention of Tony Nurenda, who did a lot of the work, particularly that straddles this area between cellular and humoral immunity as well. So it was an amazing decade. It was very formational for our kids. You can probably see from the pictures in the top half what an amazing childhood they had and what a profound impact it made on them to have grown up in Africa. And it made an enormous impact on me. I had a really vibrant, exciting time in the very, very early days of MLW. This was back when MLW was 50 people. We're now 700, so we're about a 12th the size of what we are now. But it was my first taste of what it is like to be a clinical academic and have that mixture, which I think Henry referred to, of working in medical practice in the hospital in Queens, taking what you see and developing it into a research project to address important questions with your own research team and your own data, and then finally passing some of that on by teaching and training the upcoming generation. And it was a wonderful, wonderful time. And an amazing opportunity that's given to us by the Wellcome Trust here. I just wanted to take a moment to highlight that this is the exact stage of career that we need to try and replicate for our junior clinicians in Malawi now. And this is one of the main focuses and targets for the Creator Building, is to allow our junior doctors both to develop specialist training in a clinical discipline, but also to be trained in research methods in the same way that I was, and to support a career structure that will allow our doctors to become clinical academics in this way. It's very important for MLW, it's very important for Malawi that we have clinical academics coming through. The time in Malawi came to an end. Money ran out. My fellowship ended. The Wellcome Trust didn't give us any more. And also I had to go back to the UK to complete my clinical training as a gastroenterologist. So in 2005 we made the decision to move back to the UK. I finished my specialty training, and at that point I made the decision that I wanted to become a proper grown-up research academic, which meant working through a series of posts as clinical lecturer, senior clinical lecturer and reader in the University of Liverpool. And around this time, I also started to pick up leadership responsibilities, particularly in training other junior researchers. So I was Deputy Director of the Wellcome Trust PhD programme, which you can imagine I felt very strongly about, having had such a wonderful experience myself. I helped to set up and run the Malawi-Liverpool joint PhD programme, which I think a number of people in the room have probably been enrolled on. And I also organised a Nuffield Foundation student exchange between the College of Medicine and the University of Liverpool. And I picked up two new very important mentors and guides in David Lalu and Rob Heidemann at this time as well. And as I settled into an academic career, I realised that what I needed to do was focus on getting NTS through to a vaccine. These are the kind of steps that you need really to move to that stage. You need to understand the epidemiology, you need to understand the host and the pathogen and the way they interact. You need to have a vaccine design that's appropriate. You have to go through vaccine trials, phase 1, 2, 3, and then you have to move to implementation and measure the impact of that vaccine. I was in the very fortunate position to have some fantastic scientists who had trodden this path ahead of me at MLW for rotavirus, that would be Nigel, and pneumococcus, that would be Stephen and Neil French, many of whom will know these guys. Many of you will know these guys. So I kind of had a template ahead of me. It was never going to be exactly the same for NTS, but it was something to aspire to, and there was a lot of expertise and help available to me. So, as I described to you, we had already done quite a lot of the important work we needed around host immunity and the way that the host interacts with the pathogen. And Tony Nurenda particularly continued to push this work forward during this decade, looking at the interaction between cellular and humoral immunity and what it takes for a child to develop protective immunity in the natural scenario, which would help us to then understand how a vaccine could work. So thank you for that, Tony. The other thing that we began to understand in this decade much better was the pathogen itself. And this was largely down to work by Chisom Msefula, Nick Feasey, who was my PhD student, and Benjamin Kamwenda more recently. So what we understood about the bug was that we were seeing our serovars appear in epidemics. So we had an epidemic of Salmonella enteritidis. This coincided with the arrival of multi-drug resistance. Then second, we see in the red an epidemic of Salmonella typhimurium, again coinciding with the emergence of multi-drug resistance. And when we sequenced these strains, what we found was that we were seeing special micro-adapted strains that had emerged in the setting of Africa, where we had large susceptible populations of children and adults. And there was something distinctive about these strains genotypically and phenotypically. And it was not only the antibiotic resistance, although that played a major role. So here as an example is the emergence of ST313 strains of Salmonella typhimurium. We were able to use their genotypes to map the emergence of new lineages and clades across within Malawi, but also right across Africa, and to track the way that they had followed the HIV epidemic across the continent. We were also able to use the genomes in other ways. They helped us to understand the human pathogenesis. So on the top here is a figure illustrating how when patients have recurrence, you can prove genotypically that it's the same strain coming back over and over again to within one SNP in the same bug. And Jay Hinton has done some lovely work that shows a single promoter-based change that confers serum complement resistance in our African clades, allowing them to survive in the bloodstream longer than European strains. We could also use the genomes in more epidemiological studies to trace their environmental reservoirs and transmissions in the household and between humans and adults. And we found that they have a very unique prophage repertoire, including one very highly expressed prophage, BTP1, which we suspect may be linked in some way to environmental transmission as well, and that's ongoing work from Sharon Owen. The last piece of important work that we were able to do in this time was to use a mathematical model to make the first global burden estimate for invasive NTS disease. This has since been superseded by others that have more bloodstream data at their disposal. This was a very, very important piece of work that allowed us to put NTS on the stage and make it a higher funding priority. So by 2018, we really are poised for vaccines with NTS. We've gathered all this information that we needed. There are now 2 NTS vaccines which are going first in human this year, an LPS flagellin conjugate and the Gemma membrane-based vaccine. We are part of a consortium that is doing first in human trials of the Gemma-based vaccine, and also hope to work in immunogenicity work with the LPS flagellin. So it's a very exciting time for us. We work in an environment where we have a large number of susceptible individuals, and we are in a good position to optimize the immune response of individuals to this vaccine, because you can vaccinate all the healthy children you like, but you won't actually prevent many cases of NTS because it happens in immunosuppressed children. So optimization of vaccines is very important, and we will be moving towards phase 1 and 2A clinical trials in Africa and at MLW. So very exciting times. However, in 2012, we had a surprising new typhoid epidemic. There was a big new kid on the block, and we saw the emergence of typhoid. Now, we'd had very little typhoid in Malawi for a long time going back in our blood culture records, and suddenly we saw an epidemic of multi-drug resistant typhoid. And Nick Feasey was able to lead a lot of the work describing this. So we collaborated with the Sangha, were able to show that this was an H58 multi-drug resistant strain that had jumped across from Asia into Africa on multiple occasions and then transmitted itself across Eastern and Central and Southern Africa. And when we looked at the genotype here on the right, you can see the Malawi clade emerging right at the tip of the evolutionary process. So we had some very important strains on our hand, and Nick was able to very clearly dissect out the emergence of these multi-drug resistant H58 clades within Blantyre in our own setting. A very nice piece of work that he did there. This all coincided, of course, with a moment in time where our kids had all left home, they'd headed off to university, and we were in a position to move back to Malawi. So our last big decision of the last decade was to move back here. And it was a very exciting time to be moving back because everything was primed for both NTS and typhoid work to move forward. So as I moved back, we were able to get funding from both the Wellcome Trust through a strategic award, which was co-funded with Bill and Melinda Gates. I think this was the first time that they had co-funded a large project together like this, to put together a consortium, an alliance, to try and look at typhoid across Africa and Asia. This was very timely because there's a new typhoid conjugate vaccine up and ready to go, and that just happened to coincide with the emergence of this disease in Africa. So timing here was absolutely critical. We had 2 centres in Asia, in Bangladesh and Nepal, and we were the only Africa centre. So a very important study really for the whole continent, because we know that typhoid was spreading across. And so I was able to set up this strata study, which many of you will have heard of, some of you will have been involved in. We censused nearly 100,000 people in Indirandi. We did 3 years of enhanced passive surveillance using blood culture at Queen's and out in the community, achieving 200,000 years of person follow-up. We did healthcare utilisation surveys in order to be able to correct for people who didn't show up to the hospital. We did large serosurveys. We took 15,000 blood samples in people's homes in order to be able to try and estimate not only clinical disease, but serological conversion and exposure to the pathogen as well. We were able to try to detect chronic carriers, and then to make a model of what was the contribution to transmission of acute illness compared to chronic carriage, and therefore to be able to model what would be the impact of a vaccine in these populations. Because a vaccine prevents acute cases, it prevents transmission from case to case, but a vaccine doesn't prevent chronic carriage. The good news from our model is that it seems that certainly in Blantyre, chronic carriage plays a very small part in transmission. Most of the transmission appears to be from case to case to case, which gives a very good hope that a vaccine will be extremely effective in controlling typhoid very rapidly. As part of our surveillance, we found an unexpectedly high community burden of typhoid. We found between 4 and 5 times as much typhoid when we went out to the health centre and did blood cultures as when we sat in Queen's and waited for them to come to us. So an enormously higher and undiscovered burden than we had expected, including right down to under 2s, which of course is very important to know for a future vaccine programme. And this is us using the wonderful EPAL tool that Liz Corbett's team has developed, and which many other studies now use. And this is my fabulous census team for Strata. Things moved very quickly from this point. We were able to take our Strata data even before the study had finished to the WHO SAGE group, the working group there. We presented the data, and they made a policy recommendation for typhoid vaccination from the age of 9 months, 6 months even, 6 or 9 months, right through to the age of 15 months, and pre-qualified the vaccine in November 2017. We approached the Bill and Melinda Gates Foundation together with CVD Baltimore and put together a typhoid consortium in order to accelerate typhoid vaccines forward. Gavi committed $82 million of funding towards typhoid vaccination and opened windows for applications for funding for individual countries for national introduction, and path dedicated to support countries make those applications. And as I speak, the Malawi NITAG have seen our data. They are putting together an application to Gavi, considering putting an application to Gavi towards funding, and we're hoping that that application will go in January 2019. Our major part in this consortium was to run the first, and at the time the only, clinical trial in Africa of the typhoid conjugate vaccine. Trial called TYVAC. You've probably all seen the teams out. This is my fantastic vaccination team and our core TYVAC management team sitting. We have a thing about socks in the TYVAC team. We celebrate successes with new socks. So this is them showing off a particularly fine set of new socks when we finished vaccinating the kids. We launched the vaccine trial in 26th February 2018. Golden Kondowe, a 4-year-old, was our first child. We were vaccinating children from the age of 9 months right through to 12 years. And we went on to vaccinate 28,050 children in the space of 7 months in a population of 180,000 children in Durandi and in Zingwangwa. This equated to 200 children vaccinated a day, all with individual randomization, individual consent, and individual assent for children over the age of 8. And we aim to keep them under enhanced passive surveillance and mapping for the next 3 years. It was an enormous undertaking. It was 12 times larger than the next largest vaccine trial that MLW had ever done. We had a team at a maximum of 150 people. We were working in 3 schools simultaneously every day. It was really, really a colossal effort, and I think it would be a good moment, since I hope we have plenty of our operations teams here, to thank them for the absolutely extraordinary support that they gave us during this enormous undertaking. So just to give you a short update on the study, the surveillance is working very well. We estimated that we needed to detect at least 30 cases of typhoid in the trial to know that we would have statistical power to show a significant effect. And we've already doubled that. We have more than 60 cases. We're able to map them. This map shows the kind of lime green. Every dot is one vaccinated child in the trial, and every black dot is a typhoid case. So you can see our 2 vaccine areas, Durandi in the north and Zingwangwa in the south. You can see that we're seeing typhoid cases across our whole study area. But we did have the impression that we were seeing, if anything, more typhoid in Zingwangwa than in Durandi. And because of this fantastic EPAL technology that we are able to look down to the level of which school and which catchment area we're in, and it is indeed seems to be the case that we have a 4-fold higher incidence of typhoid in Zingwangwa as we do in Durandi at the moment. This suggests to me that we are probably actually in the middle of a small outbreak in Zingwangwa, and because we're seeing very good numbers, we may be able to get some very interesting subgroup analyses comparing in Durandi with Zingwangwa, an area that's now effectively endemic, compared to an area that might be epidemic at the moment. And again, the very large number of cases that we're seeing will suggest that we may be able to get age-stratified efficacy for the vaccine. So the trial is going very well, thanks to our fabulous passive surveillance teams. I hope some of them are here. Is there anyone from the passive surveillance team here? Thank you. Great work. We did a small efficacy and immunogenicity study nested in the larger study. We have had rigorous safety review by the DSMB, by NHSRC and IRBs in the UK and the USA, and PMPB within Malawi. And the data have all now been reviewed by the WHO Global Advisory Committee on Vaccine Safety. It has a very low rate of very mild adverse events, and we have all together, I think, nearly 200,000 person years of follow-up between the 3 sites, and there is no concern at all about any signals of serious adverse events or other concerns about safety profile for this vaccine. So it's a very safe vaccine. It's very good. The trial is working. We've looked at antibody production in the first 28 days at all 3 sites. We see very good antibody responses in every age group that we test, including the very young children, which is important compared to our control children. And our sister site in Nepal has done an interim analysis after their first year, and their interim analysis shows, when you consider all ages lumped together for that one site in Asia, that the vaccine is likely to be efficacious. So we're very optimistic about this study. So this makes Malawi a really, really exciting time and place to be. We have reached a certain point with NTS where we're just about ready to move into trials. And because of this typhoid vaccine trial, we are now kind of catapulted forward. We have the experience of working with these trials, and we have a lot of the infrastructure, so that we'll get a lot of additional leverage as we try to move forward with NTS, developing NTS vaccines as well, both at a scientific level and at an operational level. I think you can see that the timing for me coming back to Malawi at this point has really been a productive time. It's meant that we've really been able to be productive, a lot of grant income, and look at the number of people in the teams. There's loads of them sitting in the back here, and I've had just amazing teams working on this. So it's a real moment of synergistic opportunity at MLW at this time. We have a picture now where we have both typhoid and invasive non-typhoidal salmonella, NTS disease. 2 different groups of susceptible individuals, one in healthy people, one in immunocompromised people, and an enormous burden of vaccine preventable illnesses and deaths that we are incredibly well positioned to prevent. And that is what I went to the NIHR with, was to try and explain to them the opportunities that we have here to make an enormous impact on both of those diseases. So I put forward to them this proposal that we would have 3 work streams, one around epidemiology, one around host immunity and the way the host interacts with the bug, and one around vaccination and vaccine trials, and that this work would go forward both for typhoid and NTS and leverage extra value from both of those. I was awarded £4 million. That will cover 7 new posts that will be supported in matched funding by NIHR and the University of Liverpool. It's an amazing opportunity. What they are expecting us to do is to move both NTS and typhoid vaccines forward. So typhoid, we will be moving forward through demonstrating vaccine efficacy. We hope to extend our follow-up from 2 years to 5 years, national introduction, and then trying to measure the impact nationally, both in terms of disease, but also of preventing the further emergence of antimicrobial resistance and overuse of antibiotics. And in NTS, we hope that this will pump prime our ability to test the new vaccine in different age groups, to assess its safety and tolerance, to look at immunogenicity, particularly in those susceptible groups that we've talked about, and hopefully to move towards efficacy trials here as well. So those are big aims, they're really big aims. Just to kind of move back to the personal for a moment, I've had extraordinary support, really, to get here from, I call it a cast of thousands. These are some of the most important people who have supported me, and I'm immensely grateful to them. I don't think I would have made it with any of them missing. I would likely not have made it here. However, you don't have to be a genius to spot that they are all men. I love them dearly. They didn't get any choice when they were born either, did they? But it leaves me kind of asking, where were all the women? And the answer is that they weren't there. I went through my career with, I'd never had a female boss as a clinician. I have never had a female supervisor or boss as an academic. By the time I was a gastroenterology registrar, my colleagues were all men. When I became a gastroenterology consultant, I was in a department of 14 consultants, and I was the only woman. So they really weren't there. It's not that I didn't pick them, and it's not that I didn't say thank you to them. They were almost completely absent. And so you can imagine it is a particular joy to me that I have such a fantastic number of female trainees and students training for various academic and clinical qualifications. And also an enormous joy and very exciting for me that we now have such strong female leadership in the University of Liverpool. And it was very exciting and inspiring for all of us, I think, to be invited to invite and be visited by Professor Dame Janet Beer, Louise Kenny and Hazel Scott from the University of Liverpool earlier this year to visit our research group and to visit MLW. We had a wonderful time with them. And I think I probably need to give Louise a particular shout out at this time, because it was Louise who more or less arm twisted me and then bundled me over the line into applying for this fellowship. If Louise ever asks any of you to do anything, give up immediately. Don't resist. It's easier just to do it, honestly. She is a very, very determined ally, a very determined supporter, and a wonderful mentor and quite an inspiration. So thank you to Louise for pushing me over the line and convincing me that I really should apply for this professorship. This extends to my daughters, actually, to make it even more personal. My two daughters are both just doing their first pieces of research now. And you'll imagine how delighted I am that they have both found themselves being supervised by women. My daughter Anna is a medical student in Sheffield, and she's being supervised in public health by Caroline Mitchell. And my daughter Miriam is working as an economist in health economics and WHO, and is being supervised by Dr. Fatima Al-Awa. And that's a very big joy to me. And as I said earlier, I think these two have probably been my biggest female supporters over the years. I would like to just close with broader thanks. I haven't mentioned here any of my international collaborators, because I really wanted to focus this around you guys and the support that I have had here. So many thanks to our study participants, to the study teams. Some of you are here, a few of you go back a long, long way with me. Many thanks to my students. A big thanks, as I said earlier, particularly to the MLW operational staff. I think the Strata and TIVAC studies really make it clear how much we lean on you and how much we need our robust processes and how essential you are, so thank you. Thank you to my fantastic mentors and supervisors, and I realised as I was putting this together, there are 4 generations of my family in there, so thank you to all of them. And last of all, I would like to thank you all in advance and in anticipation for all the help that I'm going to rely on you for this big task that we have undertaken. If nothing else, to go back to the beginning of my talk, these studies will all need interesting names. And I think it's well documented that I'm not good at that. So at the very least, you can help me come up with names. Thank you very much.